Hello, my name is Shawn Patel. My colleague Hitesh Patel and I will be presenting a scientific review of an article related to vision. This review is for our BIOL 4752 Intro Neuroscience class at the Georgia Institute of Technology. Our professor is Dr. Steve Potter. The scientific article that we will be reviewing is Spatial View Cells in the Primate Hippocampus, Effects of Removal of View Details by Robert Robertson, Edmund Rolls, and Pierre Georges Francois. Prior research with monkeys has shown that the hippocampus and fornix are involved with learning and remembering where objects have been seen. This type of memory association was further investigated with single neuron recordings in the hippocampus of monkeys while they engaged in object-placed memory tasks. One main observation was that about 10% of the neurons in the hippocampus were engaged when images were shown in certain positions. The main conclusion from the study was that spatial representation was allocentric. The term allocentric means in reference to the world and not relative to the body, or egocentric. Research that expounded on this led to the discovery of spatial view cells. The main premise of spatial view cells is that these cells portray spatial representation independently from where a hypothetical person is. These cells respond when that hypothetical person looks at a particular place in the spatial environment, and the response is independent of the location or orientation of the person. The role of the spatial view cells is thought to engage a memory system to it also, in which objects are remembered in a spatial context. The main premise here is that the primate brain has a spatial memory component involved in object memory. The purpose of the study presented in the article was to understand the amount of detail in spatial view is necessary for primate hippocampal spatial cells to respond. To carry out the study, microelectrodes were attached to two rhesus macaques. The monkeys were placed onto a specialized chair that adjusted the position of the chair to the head position of the monkey. The monkeys were allowed to roam around a 4x4 space with cups of food that were placed in the corners to subdue the monkeys into learning about the spatial environment to get the food. Eye position of the monkeys was also measured using the search coil technique. The head direction and position of the room were measured with a tracking apparatus on a specialized chair. A special program was also written to conjoin data from the microelectrodes and the positional data from the chair every time a particular cell fired. 352 cells were targeted, but only 27 cells were described to be spatial view cells. Once a spatial view cell was recognized and isolated, curtains would fall to block parts of the room to reanalyze that particular cell. 3 experiments were conducted. The first experiment looked into the firing rate of a neuron when the chair and the monkey were stationary looking at a specific direction. The second experiment, the head direction of the monkey was confined to only one direction, and the firing rate was measured of the associated cell. The third experiment looked into the firing rate of the hippocampal cells as the monkey was moving around. Afterwards, statistical tests were performed to see if the data was statistically significant. Also, X-ray radiography was performed to find the position of the microelectrodes and ultimately the position of the associated cells.
The results from the first type of experiment, in which the monkey was in a particular area of the environment and facing a particular direction in a stationary position, revealed that the firing rate of the spatial view cell was significantly higher when the monkey faced corner number 4. One way to make sure that it was not mistaken for a place cell was that the same cell fired regardless of the location of the monkey in the environment. Another thing they observed from this first type of experiment was that the spatial view neurons still fired even when corner 4 was completely obscured by a black curtain. The firing rate was not as high as it would have been were it not obscured. This supports the fact that view details are not important for spatial view neurons to fire. The results from the second type of experiment, in which the monkey was positioned in one place in the environment and was allowed to look around freely, showed that it wasn't important for the monkey to concentrate on details of the visual scene, but more so on part of the space being viewed by the monkey. The spatial view neurons that fired when the monkey looked at a certain area when the walls were not hidden still fired even when the walls were obscured and when the monkey looked in approximately the same location. From the experiment, they observed that there was a slight drift in the spatial field when the wall was obscured. However, they noticed that once the wall was revealed again, that spatial field was corrected to its proper location. The results from the third type of experiment, in which the monkey was allowed to walk around the environment freely, showed that the spatial view neurons could be located in two distinctly different cell regions. The first region was classified as the CA3 region, and they were special in that spatial view cells in this region had very low firing rates when the lights were turned off, and the monkey could not see. When the lights were turned on, the firing rates of the CA3 cells began to increase as well. The other region, which was called the non-CA3 region since it wasn't found in the CA3 region, revealed that the cells in this did not have a lower firing rate when the view details were obscured. These cells may use other cues to help them properly fire, such as the vestibular system and the proprioceptive cues. One possible theory is that the spatial view cells found in the non-CA3 regions are the ones triggered by memory when trying to recall places and locations. One reason why primates have special view cells while rats do not is that we have highly developed visual systems while rats rely mainly on their olfactory systems. Also, spatial view neurons enable us to explore and remember information about what's present at places in the environment without actually visiting those places. Primates often use these cells in the wild when they try to remember where they saw ripe fruit. And we might actually use these cells at home when we try to remember where we left our keys.